So far, we have covered the first two steps in the biological process of germination, which are water imbibition and enzyme activation. Under the topic of enzyme activation, we have looked at how stored reserves in the seeds, in the form of carbohydrates, lipids, proteins, and minerals, are broken down. In this lecture, we will go over the remaining steps in seed germination, namely initiation of embryo growth, rupture of seed coat, and seedling emergence. Before we proceed, let's revisit this slide illustrating the three phases of water uptake in germinating seeds. By now, you should all be familiar with these patterns of water uptake, as well as the key events that occur during each phase. Recall that phase 1 is defined by a rapid water uptake by the dry mature seed. This phase is also known as the imbibition phase. Phase 2 is characterized by a leveling off of water uptake as the seeds start using water in its metabolic activities. These metabolic activities include enzyme activation that degrades or breaks down storage reserves that provide food for the growing embryo. With the breakdown of the seed reserves, the growth of the embryo is also initiated. The initiation of embryo growth is characterized by a marked decrease in the dry weight of the endosperm and an increase in the dry weight of the embryonic axis. The reduction in the weight, as well as in the chemical composition of endosperm, are due to the catabolism or breakdown of stored carbohydrates, proteins, lipids, and minerals in the endosperm and their subsequent transport to the growing embryo. The increased respiration that results in leakage of nutrients from the imbibed seeds also contributes to dry weight loss. Let's take a look at the second panel here that shows the changes in the dry weight of the different parts of a corn seed during initiation of embryo growth. Focus your attention on the changes in the dry weight of the endosperm, which is this line here, as well as of the embryonic axis, which is this line here. The x-axis in this graph indicates time, while the y-axis indicates weight or composition expressed in milligrams per part. So as you can see, with time, the dry weight of the endosperm decreased, while that of the embryonic axis increased. The same pattern can be observed with total nitrogen, as well as the insoluble and soluble proteins. Again, the observed decrease in the dry weight and chemical composition of the endosperm are due to the breakdown of the stored carbohydrates, proteins, lipids, and minerals, and their transport to the growing embryo. Here are more graphs depicting changes in the content of nucleic acids, carbohydrates, amino acids, and fats in germinating seeds. Look at the level of these chemical components and notice how they are higher in the embryonic axis compared to the endosperm. Even the fats and oils are increasing in the embryo axis, as you can see here. The endosperm of corn seeds is composed mostly of carbohydrates and proteins and not a lot of fats or oils. The fats are mostly found in the scutellum, which is considered a modified cotyledon in monocot seeds. Now look at the fat content in the scutellum. It is also decreasing, while the fat in the embryo axis is gradually increasing. Again, this is due to the breakdown of all the reserves in the seed and their subsequent transport to the embryo. With a continuous supply of energy to the embryonic axis, naturally, we will see a progression in the growth of the embryo. Between phase 2 and phase 3 of water uptake, the radicle or the embryonic root emerges. This root elongates and becomes functional. The functionality of the root brings the seed to phase 3 of water uptake. The developing roots aid in the increased water uptake by the seed during this phase. The emergence of the root out of the seed coat signals that germination is complete, and that's the very basic characteristic of a germinated seed, when the radicle or the embryonic root emerges from the seed coat. Now, radical emergence is accomplished by both cell elongation as well as cell division. In general, the emergence of the radicle through the seed is initiated by cell elongation followed by cell division. This is an illustration of a root showing different cell activities. The root tip is a site of cell division, and that makes sense because the root needs to elongate, and that can be accomplished by cell division. Slightly above the root meristem or root tip is a site of cell elongation, and above that is a site of cell differentiation. Cell differentiation is important in the development of other organs, such as lateral roots. Now let's go back to the site of cell elongation. Cell elongation, as it relates to radical protrusion, proceeds into two stages. The first stage is characterized by slow radical elongation without any decrease in dry weight and only a small increase in fresh weight. The second stage is characterized by a rapid radical elongation marked by an increase in both dry and fresh weight and accompanied by rapid nutrient mobilization. Following radical protrusion is seedling establishment. Seedling establishment is actually no longer a part or a key step in germination. As I said earlier, germination is technically defined by the protrusion of the radical or embryonic root from the seed coat. This means that seedling establishment defines the beginning of a totally different phase in the growth and development of a plant. Seedling establishment is defined by the rapid growth of the shoots immediately following radical protrusion. During seedling establishment, the roots of the seedling move downward while shoots move upward in a gravitropic response that is mediated by the plant growth hormone known as auxin. The new seedling transitions from being dependent on the food reserves in the seed to producing its own food. As the seedling becomes firmly established in the soil, it begins water uptake and photosynthesis, and so begins another cycle in the plant life. The biological process of germination starts with water imbibition, followed by enzyme activation, embryo growth initiation, radical protrusion, and seedling establishment. Enzyme activation is important for the breakdown of stored reserves, including carbohydrates, lipids, proteins, and minerals. The products of the metabolism of these reserves will provide a growing embryo a source of energy. Embryo growth initiation is characterized by a marked decrease in the dry weight of the endosperm and an increase in the dry weight of the embryo axis. The reduction in the weight and chemical composition of endosperm are due to the catabolism or breakdown of stored carbohydrates, proteins, lipids, and minerals in the endosperm and their subsequent transport to the growing embryo. With a continuous supply of energy to the embryonic axis, the radical or the embryonic root finally emerges. Radical protrusion signals the completion of germination. With the continuous development of the root, the seedling gets established in the ground. Seedling establishment is a process that allows the new seedling to process its own food via water uptake and photosynthesis and begin another life cycle.